Hi, my name is Jennifer Lee, and today I will be presenting on my project, the identification of stomata-related tDNA mutants in Arabidopsis thaliana. So what are stomata? Stomata regulate gas exchange, and they consist of two guard cells and a pore in between, and these stomata are formed via a series of symmetric and asymmetric divisions. Proper patterning of stomata, which means that there's at least one non-stomata cell in between two stomata, leads to efficient gas exchange. So the pathway starts with the binding of EPF2 or EPF1 to one of the members of the ER family, which works in conjunction with TMM. Binding of this ligand to the receptors triggers a MAPK cascade, which then inhibits these positive regulators of stomata, as in they regulate the symmetric and asymmetric divisions, leading to the development of mature stomata. So what is tDNA? tDNA stands for transfer DNA, and it is a transfer of a genome between a bacterium and a host plant. These are genetically and physically stable. There's a discernible effect on gene expression, and there's a 1.5 to 2 insertions per line that are randomly distributed. Taken together, I plan to genotype EPF1 mutants and as well as genetically identify stomata-related tDNA mutants. To this, I have two approaches. There's the genetic approach. Mutants with no stomata are characterized with smaller and more yellow leaves. Mutants with excess stomata are harder to discern from wild-type plants, since they will appear greener and healthier than plants with no stomata, and we'll have to use a microscope to distinguish between these mutants as well as the wild type. Approach number 2 is the molecular approach, where we will genotypically confirm the tDNA insertion through PCR, and this is a definite test if the mutants do not show a phenotype under the confocal. So we'll start with TMM, and since TMM and ER work together, their phenotypes are pretty similar. So we can see here that there's a violation of this one cell spacing rule, where there's not a non-stomata cell in between stomata. We can see that in here and here. Going down to SCREAM-D. SCREAM-D isn't a transcription factor, but it modulates the activity of the transcription factors by forming a dimer with them. Mutations in SCREAM-D lead to overproliferation of stomata, leading to the entire epidermis of the leaf being covered by stomata. And as I mentioned before, these are hard to discern with the naked eye of which plants are mutants and which are not. So we would have to use the confocal to examine the epidermis of the leaf up close. The first transcription factor is speechless, regulates the transition between a mother meristemmoid cell into a meristemmoid, so therefore it regulates the entry into the stomata lineage line. And since mutations in this particular transcription factor can prevent the lack of stomata or any of the successive divisions. Next transcription factor is mute, and these small cells are arrested meristemmoid, which is a stomata precursor cell. And since mute regulates the transition between meristemmoid and a guard mother cell, these mutations in mute would lead to these small cells unable to differentiate into guard mother cells. And then there's FAMA, which is the last transcription factor, and it regulates the transition between the guard mother cell to the guard cell. And we can see that there are divisions happening, but none of these divisions will lead to a formation of the proper guard cell. So as for the tDNA insertions, these are the names of the mutants I'll be analyzing, as well as their putative insertion sites within the peptide ligand EPF1, which regulates the spacing between stomata. This is a representative gel of a line called SOC 035606. Since the insertion is so large, regular wild-type primers will not be able to replicate the DNA through PCR efficiently. However, with the addition of mutant primers, particularly LBB 1.3 on the left border, the DNA will be able to be replicated if there is an insertion within that line, and there is an insertion within that line. As we can see here, there's a band around the 1 kilo base pair region. 1 and 11 are the controls. Unfortunately, the control didn't work out. There's supposed to be a band for the wild type under the control, so unfortunately we cannot conclude that this plant is homozygous for the gene, but we can at least conclude it's either heterozygous or homozygous. So once we discern the problem with what's happening on the molecular level, I plan to assess the mRNA expression of soft tDNA mutants using RT-PCR, as well as determine the phenotype of those mutants using the confocal microscope. Thank you for listening to my presentation, and have a nice day. Thank you.